friends. So today we are again here with another important topic. Today we'll discuss about the history of DNA, some of the relevant questions which are asked in the examination, coming up with the questions as well as with a brief explanation. So first question, which of the following statement is false about DNA? You have to select the false statement about DNA. So you have four choices. First of all, you clearly read the choices. Is it located in chromosomes? Carries genetic information from parent to offspring? Abundantly found in cytoplasm? There is a precise correlation between the amount of DNA and number of sets of chromosome per cell. So the false statement is it's abundantly found in cytoplasm. So to get a clarity about the answer which we have marked, we'll give you a brief explanation. So we all know that in case of eukaryotes, the DNA, it is found in nucleoplasm. So this nucleoplasm, it is surrounded by the nuclear membrane. So the DNA, it is found in the nuclear membrane. This structure is known as nucleus. This whole combined structure, this we call it as nucleus. This is found in the cytoplasmic matrix. So this entire thing is in case of eukaryotes, while in case of prokaryotes, we know that DNA is found in a less dense cytoplasmic matrix, and this is known as the nucleoid. So I hope this explanation is fine for you. Coming up to the next question. Which of the following function of DNA is necessary for the purpose of evolution? So for the purpose of evolution, what is indispensable? It is replication, transcription, translation, or mutation. As we all know, if there is mutation, it will lead to evolution. So a brief explanation about it. Mutation facilitates the change of bases within a DNA, and if this change encodes for a viable amino acid. So there is a change of bases, and if it results in change of viable amino acid, so which in turn may lead to the synthesis of a different protein. So this will result in the formation of a different protein. This protein, it exerts a phenotypic character to the organism which may be different from the wild type. So this whole process, as you know, if there is a change of bases, it will if it results in a different viable amino acid, then different protein is formed. It results in a different phenotypic character which may result which is different from the wild type character. So this finally it will lead to formation of a unique character itself, and this is what it will finally lead to evolution. Next question. According to the phenotypic characters of pneumococcus considered in Griffith's experiment of transformation, we know that this transformation experiment, it was done by Griffith. So the name of the scientist, it is also asked in many of the papers. Kindly noted down this transformation experiment, by whom it was done. Which of the following statements are correct? You have to choose the correct statements from the following. The correct answer is, you can see it's C. This is, so you have to, this one is the correct option. Next, this one is the correct option, and the fifth one is the correct option. So we'll look at all the options. So what is Griffith experiment? So in this presence of slime layer, as we all know, the slime layer was not considered. So here, this experiment, the major part was the presence of capsule or the absence of capsule, which resulted in smooth and the S and the R type. Then it was the molecular makeup of the capsule, which was able to differentiate. So coming up to its explanation. As the two phenotypic characteristics of importance in Griffith experiment. So what was important? It was the presence or absence of polysaccharide capsule that surrounds the bacterial cell, which is responsible for its characteristics smooth or rough colonies respectively, S and R colonies. So the type of capsule, that is the specific molecular composition of polysaccharide. This is the molecular composition of the polysaccharide which is present in the capsule, coded by its genome, thus triggering immune response of a specific type. So this was the presence or absence of a polysaccharide capsule which is responsible for its smooth or rough colonies, and this capsule, it has a specific molecular composition of the polysaccharide present in the capsule coded by its genome. So this resulted in a different immune response. This entire experiment, it was, so the correct options as we have selected, this is presence of capsule, absence of capsule, molecular makeup of the capsule. Next again the question is on Griffith's experiment. Which of the following combination is correct observation for transformation experiment performed by Griffith? So we have to select the correct option out of this. Type 3 is living plus mouse is equal to dead. So type 3 is heat killed plus mouse is equal to dead, or type 2 are living plus mouse is equal to dead. Which is the correct option? As we found that instead of moving on to the next options, we found that A is already the correct options, and here comes a brief explanation about it. So type 3 strains, it is the smooth strain and it is virulent in nature. We all know that this one S, as previously we have discussed that the capsule having this smooth strain of pneumococci, which is virulent in nature. If this strain is injected, now this strain, the virulent strain, it is injected in alive in a mouse. It is bound to develop the disease and thus fall dead. So as we all know, rest of the options are not correct. Here it is healed killed, so it will not be virulent. Next, R type, that is not virulent. Then again the heat killed is there. So this one is the correct option. Number 5. Frederick Griffith's experiment involving Streptococcus pneumonia lead to the discovery of. So here are the options. DNA as a genetic material, RNA as a genetic material, protein as a genetic material, transforming principle. We all these questions are interrelated. You can see previously also we are going through the same questions time and again, and so here also in the previous all these questions are interlinked. So it is a transforming principle. So here you will get again a brief discussion and a clear discussion about this. All the previous also will be discussed. So when heat killed virulent smooth type bacteria were injected in the mouse along with living avirulent rough type of bacteria, the mouse developed the disease, which was an unlikely result. So this was heat killed and it was injected along with the living avirulent rough type, and the mouse it developed the disease. This was an unlikely result. Moreover, when the bacteria were isolated from the infected mouse, they were found to be of the virulent type. So, this is the S type. This was the virulent type. It was uh, injected in the mouse along with the living avirulent type, heat killed, plus living avirulent type, and the mouse it developed the disease. And what we found out is mouse they were found to be virulent type. Thus, it was believed that some factor from the heat killed virulent type bacteria transformed. So here took the transformation, transformed the avirulent to the virulent type. The avirulent type was the rough type. So it was finally the transformation took place from the heat killed to the avirulent type bacteria, which was known to be as the transforming principle. So I hope this is clear to you. So what exactly is the transforming principle? Next, define results proving DNA to be genetic material was given by. So who proved this that DNA is the genetic material? This was done by, was it Griffith? Griffith, as we all know, discussed previously, it was related to the transformation experiment. Was it Hershey and Chase? The correct option is C. Avery, MacLeod and McCarthy. This is a very important question and it has been asked many times. Please commit it to your memory. So these were the three scientists, Avery, MacLeod and McCarthy. In the experiment with pneumococcus strains, type 3S and type 2R, they performed three separate experimental setups. So, the three experimental setups they performed, this was as under: using DNAs to degrade DNA, using RNAs to degrade RNA, and using protease to degrade proteins. Thus, when the separate combinations were injected into mice, respectively, the transforming principle was found to be DNA. What exactly? This transforming principle, it was found to be DNA. Only these three enzymes. So these three enzymes we all know: DNAs, RNAs and protease. As only their subsequent substrates were the main factors that were in conflict to be the genetic material. Hence, it was proved by them DNA was the genetic material. These three people, they proved it. Next, which of the following statements regarding the mechanism of transformation in Bacillus subtilis is false? Here we have to select the false statement. Kindly note it down. In a hurry, sometimes we read, we instead of going on into the details and clearly reading the sentence, we in a hurry up start marking the options. First of all, you have to be clear with this, whether they are asking for the correct option or the false option. So you have to select down the false option from this. So the answer is, a component cell contains a DNA receptor translocation complex. No, this is wrong. Then this is right. Then coming up to the next, uh, this statement is false. I'll read it in detail. While translocation of exogenous DNA, both strands get passage into the cell. This one is a false statement. Rest is also correct. While translocation of exogenous DNA, only one strand gets passage into the cell. We know this thing. So both the strands, they do not get passage into the cell. The exogenous DNA recombines and incorporates itself into the chromosome of the recipient cell. This is also correct. Then first one is also correct. So the false option is this one, B1. This is the false option. Now moving on to a brief explanation about it, how we selected this. What happens in translocation? Translocation of exogenous duplex DNA. We all know that it is DNA, it is duplex, two strands are there. One of its strands gets degraded by deoxyribonuclease, that is the DNase, in the periplasmic space, thus allowing passage to only one single stranded DNA molecule into the cell. So, I hope you are clear with it, how the translocation takes place. Coming up, next question. What were the main criteria taken under consideration for the experiment by Hershey and Chase? So here we have to select the main criteria which was undertaken. So is it DNA contains phosphorus, protein contains sulfur? This is also the main criterion. Then protein contains phosphorus. This is the wrong option. Then both DNA and protein contains phosphorus and not sulfur. Again, this is not correct. Both the DNA and protein contains sulfur and not phosphorus. So finally, having a quick look at all the options, we found that A is the correct option. This one, A part. Now, brief explanation. DNA contains phosphorus in phosphodiester linkage and do not contain any sulfur. You have to be clear with this. DNA is primarily composed of phosphate, nucleotide bases, and deoxyribose sugar. So these three components, they finally form the DNA. Whereas proteins, they are composed of sulfur containing amino acids. Number 9. Next question. Which combination of radiolabeling is correct in Hershey-Chase demonstration of DNA as genetic material in T2 bacteriophage? So out of these options, we have to select the correct option. So I find that again the A1 is the correct option. This was chosen. 31P and 35S. Rest of the options, they are not correct. So the two factors considered by Hershey and Chase were the simple composition of T2 bacteriophage, that is DNA and protein. Thus radiolabeling of only one element was required, 32 phosphorus and 35 sulfur being the two radioactive isotopes. Therefore the combination of either, so we have to choose either one combination, that is 32P, 32S or 31P and 35S. Both the options we can't choose it together. Thus radiolabeling, it requires only one element was required. Hence the correct option is 31P and 35S. Question number 10. Recently, scientists have developed a procedure in which protoplasts of E. coli could be directly infected with the phage of DNA. So, what is exactly this process is known as? Protoplast of E. coli could be directly infected with the phage of DNA. This process is known as transfection. How this process occurs? Now, in detail, we'll read about it. Transfection is the process of the introduction of exogenous, naked, and purified nucleic acid in any organism. So here, the exogenous naked DNA, it is introduced into any organism and this is known as the transfection. In case of transformation, what happens in case of transformation? The introduction of nucleic acid is via plasmid. So here plasmid or any such vector. Some vector is needed for the introduction of the nucleic acid, exogenous nucleic acid in case of transformation. Here what happens, it's the introduction into any organism. Here no vectors, and next is transduction. These three you have to be clear with these three, that is transfection, one is transformation and the other one is transduction. Transduction, how it is taking place? The process of introduction of nucleic acid via viral vector. Here what kind of vector is involved? Only it is the viral vector, and here via the plasmid or any such vector, that is the transformation. And transfection, it is again a different thing, that is introduction of exogenous naked and purified nucleic acid in any organism. Lastly, what is mutation? Mutation is the alteration of nucleic acid at its composition level. It has nothing to do with the transformation. So mutation is the alteration of nucleic acid. It has nothing to do with the DNA transfer. Next, question number 11. According to Mendel's experiment, genes carry genetic information from parents to offspring. On which plant did he perform his experiments? So Mendel experiment, which plant did he choose to carry out the experiments to prove his results? It was Pisum sativum. So we know Gregor Mendel, he performed his experiments on garden pea plants whose scientific name is garden pea plants. We know the scientific name, that is the Pisum sativum. The three scientist names, the other three scientific names, Oryza sativa, Allium cepa and Vitis vinifera, belongs to rice, onion and grapes family. So this one, Oryza sativa, it is for rice. Allium cepa, we know that is the scientific name for the onion, and Vitis vinifera, that is for grapes. So it was that Mendel, he performed his experiments on the garden pea plants, and the scientific name is Pisum sativum. Next, now a question about the plasmids. Plasmids, you have to select the correct option about it. So correct option I find about the plasmids is, after reading all the options, it's that it's circular double-stranded DNA. It's B part. It's not linear, supercoiled, double-stranded DNA. It's circular double-stranded DNA. So found mostly in prokaryotic cells. Plasmids are extrachromosomal, auto-replicating, small circular, double-stranded DNA which generally carries certain genes for the advantage of bacteria such as antibiotic resistance. So it is mostly found in the prokaryotic cells, number 1. Then there are the extrachromosomal auto-replicating, number 3. Then small circular double-stranded DNA which generally carries genes for the advantage of bacteria, that is the antibiotic resistant genes and all. Next question 13. Which stores the genetic information in DNA? So the genetic information, we are all saying that DNA is the genetic material, DNA is the genetic material. What exactly out of this stores the genetic information? So correct option is the nitrogenous base. Here comes the explanation for it. Genetic information is stored in the sequence of nitrogenous base, as they are of 4 types and they are A, T, G, C. So here comes the difference, because they are of 4 types, the nitrogenous base, and so they will decide what is the genetic information. The order of their occurrence decides the mRNA sequence. So order, it will result into a mRNA sequence which in turn codes amino acids, leading to synthesis of protein. So hence, finally we can say what exactly decides the genetic information, that is the nitrogenous base. Sugars and phosphates, they form the backbone and are common to all. So sugar and phosphate, as we all know, they are the same for all, whereas polymerase helps in the replication of DNA template. Polymerase, we know that it is the enzyme and it helps in the replication. So finally the correct option is this. Number 14. DNA always carries the genetic information for all organisms. Is this a true statement or a false statement? Answer is it is a false statement. So, why it is a false statement? Here comes the reason for it. What is it? There are certain viruses in which RNA carries the genetic information. They are known as RNA viruses. We are very clear with it. Sometimes the RNA is also a RNA, it carries the genetic information. So we can't say that DNA, it always carries the genetic information for all the organisms. This is not the correct statement. They are known as RNA viruses, for example, retroviruses, influenza viruses, many other also are there. Then 15. What is the major type of RNA produced in the cell required for translation? Is it mRNA, tRNA? So it is the ribosomal RNA. This is the abundantly found. Generally, rRNA is of 4 types. We know it is of 4 types: 28S, 5.8S, 18S and the 5S. These subunits, they comprise 80% of the total amount of RNA produced in a cell. This is the major rRNA, ribosomal RNA. This is the major and are majorly required for translation, and these rRNAs along with proteins form ribosomes which reads the codes for translation.